Hello, everybody. Welcome to the Nanomotor Mini Talks. My name is Wei Wang. I'm a university professor in Harbin Institute of Technology, Shenzhen. So I'm talking to you from Shenzhen, China. We're 9 p.m. right now. So before we begin today's talk, I'd like to give a very quick overview of why we're doing this series. Because nanomotor papers are being published on a daily basis, and we all have a very busy schedule. Sometimes we just can't read all the good papers. On the other hand, the authors of any scientific paper would love to talk to their readers and get feedback. So I thought it would be a wonderful idea to invite authors of recent papers so they could tell their own stories and maybe give us some behind the scenes stories. We plan on having at least one talk every week, starting from this week. And hopefully we do this on the same time every week, and make sure join these talks and spread the news to your friends and colleagues. And also, these talks will be recorded and later will be uploaded on YouTube. And you can search for a Nanomotor Mini Talks or a Nanomotor Update on YouTube to find these talks. All right, so today's talk is from Caleb Meredith. Caleb is a PhD student at Penn State University, and he'll be talking about his recent paper published on Nature Chemistry. Please go ahead, Caleb.
Okay, hi everyone. Good morning, good evening, good afternoon, wherever you are in the world, and thanks so much for coming out. I'm very excited to kick off this talk series and talk to you about some research that we did that was recently published, where we were observing these predator-prey-like interactions between droplets. And so I'll share with you all the story of how we came to discover this behavior, how we understand it based on the local generation and uptake of chemical gradients between these droplets, and the sort of design principles that we learned along the way that can be used to extend this type of behavior to many kinds of emulsion systems. So before I get started, I just want to begin, with my acknowledgements. So this is some research that began about 4 years ago when I started my PhD, and was completely a collaborative effort. Could not have happened without the team. I specifically want to acknowledge, though, my co-first author, Pepin Moorman, who is now a postdoc at Johns Hopkins, and my advisor, Lauren Zarzar, whom this could not have happened without. Shout out to the rest of the Zarzar lab here at Penn State, as well as the funding. All right, so let's jump in. So the inspiration for this research that we're pursuing comes directly from nature. And it's this observation that at all scales, all shapes and sizes, chemical signaling is used to create self-organization and functionality of all different sorts of creative ways. And in this video, you can see an example of a chemical signaling in the form of a chasing interaction between a white blood cell and a bacterium, where there's no brains here. It's just chemicals being exchanged between the two organisms that it's mediating this behavior. And as someone who is deeply inspired by nature, as well as is a material scientist, I would like to know whether we can create sort of synthetic materials capable of hopefully getting close to this level of sophistication in a man-made chemical system and material system. So the problem, even though these organisms are relatively small, the sort of precise mechanisms of these biological potential pathways here is still exceedingly complex. So sort of our approach is to study sort of a more minimal chemical system that could be more tractable in sort of understanding the essential dynamics for what might be your sort of essential ingredients for creating this sort of active dynamics. So our approach for that was to study emulsions. And so these are stabilized dispersions of immiscible liquids. And recently in the literature, there have been several examples of active behavior in relatively simple emulsion systems, where you have these droplets that are dissolving out of equilibrium and can become self-propelled in response to a self-generated chemical gradient that they're releasing, as what is shown in this video. And so these gradients arise spontaneously due to a process known as solubilization, whereby oil from inside these droplets partitions into surfactant micelles in the continuous phase, residing in their hydrophobic cores. And so the droplet motion arises in response to the Marangoni effect, where interfacial tension gradients across these droplets generate fluid flows that cause droplets to move in the direction of lower interfacial tension. And so these interfacial tension gradients in this case are arising in response to the chemical gradients being released by these droplets as they solubilize. And specifically in these systems, what you have is higher concentrations of oil-filled micelles result in higher interfacial tensions, meaning that the droplets are effectively repelled by their own chemical signal that they're releasing. And so above a critical threshold, a fluctuation will induce spontaneous symmetry breaking, causing these droplets to propel away from where they once were. And not only can you have self-propulsion on the sort of individual droplet level, but more recent work has shown that below the critical self-propulsion limit, you can have mutually repulsive interactions between droplets of the same chemical species. And this is due to effectively the overlap of their mutually repulsive gradients. So you have a buildup of this oil leading to higher interfacial tensions between the two droplets. So with this understanding in mind, we wanted to, where you can have interactions between neighboring droplets due to the chemicals that they're releasing, we wondered if by mixing multiple types of droplets with different chemical components or different species under the similar conditions of solubilizations, could that lead to more complex interactions and more interesting active dynamics? So the way we went about doing this was to begin with two types of oils, ethoxy nonafluorobutane and bromooctane in a Triton surfactant. And we chose these two oils because of their significant chemical difference, first of all. So one is a fluorocarbon, the other is a hydrocarbon. These oils are also mutually miscible completely, have low water solubility, and are both denser than water, so they sink, which is quite convenient for experiments. And so using microfluidics, we could then create monodispersed populations of each of these two types of droplets. And then using a pipette, we could take some droplets from each species, mix them up in a dish containing some surfactant solution, agitate it to randomly disperse them, let the droplets settle down, and then wait. And then within a few minutes, if we looked under the microscope, we would start to see these intriguing dynamics start to take place, where there's a few things I want to point out here. So one is the large population of these asymmetric droplet pairs that always swim in sort of the same orientation, with the darker bromooctane droplet in the back and the lighter EFB droplet in the front. We also see that larger clusters containing 3 or more droplets can have more complex motions, translational and rotational components that depend on the underlying symmetry of the cluster. And I'll come back to this later on in the talk. But taking a closer look at these pairs again that we noticed, if we looked closely at the lead up to the formation of the chasing pair, so that interaction beforehand, we see that they are indeed non-reciprocal, sort of like the dynamic between predator and prey, where the predator is attracted to the prey and the prey is repelled from the predator. So as the bromooctane predator approaches the prey, we see that the EFB droplets begins to move away, but it's then eventually caught, forming this pair which continues to translate in the same direction. So we we believed at first that this behavior should be caused by some chemical gradients that are being shared and experienced between these droplets. And so to sort of understand what might be causing all this, we next looked at the solubilization properties of each of these oils in the surfactant solution. And what we find really is that the bromooctane oil is solubilized in the Triton surfactant, where the EFB is not. So we have a preferential solubilization of one of the two oils by the surfactant. Furthermore, if we mix up a bunch of both of these types of droplets together in the dish and wait for long times, we also observe evidence of oil exchange occurring from bromooctane to the EFB droplets. And this is evidenced to us by changes in the refractive index of the EFB droplets over time, where its relatively low index, as it increases due to the incorporation of the bromooctane oil, eventually becomes index matched with the surrounding aqueous phase, so where the droplets turn transparent looking for a period of time and then reappear. So taken together, these two observations of preferential solubilization and oil exchange led us to propose a sort of hypothesis or a framework for the mechanism responsible for this behavior. And that's that you have an asymmetric exchange of oil between the droplets that's driving these interactions. So what I mean is that the bromooctane droplet, because it's able to be solubilized by the surfactant, is able to partition into the continuous phase. And because these two oils are miscible, as I mentioned, if you have bromooctane filled micelles nearby EFB, some of that oil will become taken up into the EFB droplet. And because the EFB oil is not solubilized, this sort of reverse pathway for exchange is not allowed. So you have sort of a selective, because the surfactant solution is sort of selectively permeable to only one of the oils, you can have this net directional exchange occurring between these two species. And so another way to think of this is that you have sort of a source sink dynamic set up where the bromooctane droplet is a chemical source acting as a chemical source into the surroundings, and then the sink, if brought nearby the source, will modify the source's gradient so that you have a lower concentration of solubilized oil in between the droplets, which is what draws the predator to the prey. However, the prey is still repelled by that predator's signal, and so together these the pair modifies the chemical gradient to allow them to move in this way. So this was our idea, and we wanted to sort of put it to the test by then quantifying the strength of these interactions as a function of different variables that we thought would affect the magnitude and direction of this oil exchange occurring between the droplets. And so to do this, we looked at these chasing interactions, so the lead up to the pair formation, under many different experimental conditions, and then using tracking software, we could measure the speed of the two droplets as a function of their separation distance. And so what we found was that you have, these interactions were occurring over a distance of tens of microns, and is that as these droplets got closer together, they would then accelerate and snap together. And however, because these interactions are non-reciprocal, we also have to consider the fact that as these two droplets are coming together, they're also translating. So there's a net displacement of the pair as they come together. And so we also have to account for the speed of the midpoint between these droplets as a function of their separation distance. And because these droplets can be treated as low Reynolds number swimmers, their swimming speed is directly proportional to their propulsive force being matched by the drag constant. And so by then integrating the speed versus distance curves, we can extract effectively energies related to the strength of these chasing interactions and their displacement. So we can then plot these energies as a function of, for example, surfactant concentration, and we find, as expected, that both the magnitude of both energies increase with increasing surfactant, as you would expect due to the increase in the number of micelles responsible for transporting the oil between the droplets. So the next thing we looked at was the effect of oil miscibility. And because we thought the exchange of oil was critical for this behavior, we wanted to see what would happen if we reduced the miscibility between the predator and prey oils. And so how we did this was by substituting the EFB oil for other oils of increasing degrees of fluorination, we could decrease the miscibility with the bromooctane. And what we found was, in the limit of low miscibility, we no longer observed these chasing interactions, which makes sense, because even though you have the bromooctane still acting as the source and releasing its oil, if you no longer have a sink that is capable of uptaking that oil, there's no reason to drive this source with the predator towards the prey, so you don't end up with a chase. What we also found here was that for partially miscible combinations of oils, we did indeed still observe chasing, implying that the necessary amount of oil required for exchange between these droplets is relatively small. So the next variable we looked at to probe this framework was by changing the surfactant type. So switching out the Triton surfactant for a fluorosurfactant Capstone FS30, we still observed chasing behavior. However, in this case, the direction of movement was reversed, such that now the EFB droplet was acting as the predator and the bromooctane is now the prey. And this actually makes sense because the Capstone surfactant preferentially solubilizes the fluorinated oil in this case. So the direction of oil exchange allowed between these droplets is effectively reversed. And what we also found here is that by blending the two surfactants such that both oils were being solubilized at appreciable rates, we no longer observed chasing behavior. And that's instead, we observed a mutual repulsion due to the fact that now both droplets are releasing these repulsive signals that would then overlap. So this sort of taken together, these series of observations made us feel confident that we sort of understood this general framework for this preferential solubilization and net exchange of oil between the droplets as what is the sort of necessary ingredients for creating this chasing effect. And so we wanted to see if we could then extend it to other types of emulsion systems besides just bromocarbons and fluorocarbons. So to do this, we investigated a series of iodoalkanes from 4 to 16 carbons, which are all liquid at room temperature, and like bromooctane and EFB, are all denser than water, which is, again, convenient for doing these experiments. And surprisingly, we found that chasing could occur between two pairs of oils with only a single carbon length difference between them. So here you see the interactions between iodoheptane chasing iodohexane. And exploring all the binary combinations between these different iodo oils, we found that chasing occurred between many of the oil pairs. And so the results of that are shown here in this matrix, where the length of oil A and B are shown along the sides, and the speed of chasing is indicated within the square, as well as the color representing whether A chases B or vice versa. And so interestingly, what we found was that the trends in the chasing behavior did not scale linearly with the length of the oil. For example, iodooctane chases both iodohexane and iodohexadecane, oils that are both shorter and longer in length. So in general, the intermediate length oils tended to chase as predators, while the shortest and longest oils tended to be prey. And so to make sense of this puzzle, we measured the solubilization rate for these oils as a function of chain length. And what we found was a non-monotonic trend in the rate with this sort of local maximum here in the intermediate range. And so looking at this and into the literature, what we realized is that there are actually two plausible mechanisms for the way that this oil from inside the droplets can go into the surrounding aqueous phase. And so for the shortest length oils, such as iodopropane and iodopentane, these oils are actually somewhat water soluble, and so the oil can just dissolve directly into the aqueous phase. Whereas for these longer oils that are more hydrophobic, they require facilitation by the micelles to partition into the solution. And so these two mechanisms of water dissolution and micelle solubilization are what you contribute to this trend in the curve. And as you continue to increase the chain length of these oils, the solubilization kinetics for both of these pathways eventually decrease. And so what this suggests is really that only the micellar solubilization pathway for these dominant for these intermediate length oils is what is responsible for the Marangoni mechanism driving the motion of these droplets. So although these shorter oils are releasing oil, it's not able to be transduced into interfacial tension gradients, which contributes to their motion of the droplets. Okay, so now I want to return to the sort of multi-body dynamics that I talked about briefly earlier. So as I mentioned, as we mix up these droplets, they start to form pairs, but over time they assemble into larger and larger clusters. And excuse me, these depending on the symmetry of these clusters and their size, they can have different components of translational, rotational, or stationary motions, which makes sense. And furthermore, what we found is that over time, over the course of experiments, as the predator droplets continue to shrink and get smaller relative to prey, is when we started to observe more sort of dynamic and transient sort of movements, such as this trochoidal or run and tumble motions, which you can see examples of in this video here, you may have noticed. And so not only can we observe these different movement dynamics, but we can actually simulate them and predict them to some degree. So by adopting a relatively simple framework put forward in the literature, by sort of setting up a distant dependent force that depends on the interaction between droplets of different types as well as the same type, you can sort of just, by studying these two-body interactions that we quantified earlier, we can extract these coefficients and plug them into a simple model, which is able to reproduce to a sufficient degree some of the experimentally observed dynamics for several droplets on the order of. And we can also look at the large-scale dynamics of hundreds or thousands of these droplets as we throw them into the dish and see what happens. And so over time, they aggregate into increasingly large clusters, which, unlike a typical aggregation process, these clusters are dynamically rearranging and fragmenting, which is a consequence of this being a far from equilibrium system. And so that would wrap up what we demonstrated in this paper. So to summarize, I've shown that solubilizing droplets are able to exhibit non-reciprocal interactions when you have this selective solubilization and asymmetric exchange of oil that can occur enabled by the surfactant and the miscibility of the two oils, and that these droplets can then assemble into clusters that exhibit very interesting and complex, but also understandable dynamics. So just to wrap here with a slide on some of the future directions we've been thinking about and working on. So we've been sort of trying to come up with other ways that we can introduce different kinds of asymmetry into the chemical gradients being produced by these droplets by increasing the chemical complexity or physical complexity of the droplets. And so one way that I've been working on with many other folks is by creating Janus droplets, as you see in this video on the left, where you have two oils, one of which is being preferentially solubilized and can lead to this same sort of directional active movement. And you can read about this in our preprint for the moment. And my other group mate, Sonic, has shown that you can wet nanoparticles to the oil-water interface of these droplets, which modifies the local solubilization properties in that region of the droplet, so that if you have a partially coated droplet with these particles, you can induce some asymmetry just by physically patterning these particles on the surface. And that's another way to enhance and affect their movement. And so that's where I'll wrap there for now. Thank you all for listening, and I'll be happy to take any questions.
Thank you, Caleb. That is a wonderful talk. Very beautiful results. Any questions or comments? You can just unmute yourself and start talking. And if you're too shy to talk, you can put your question in the chat box.
Sorry, I just joined in the middle, so I have a question regarding the droplet size.
Yes.
What's the size regarding the bright droplets and the big gray droplets?
Yeah. So the size of the droplets is on the order of tens of microns. Typically, we use droplets that had a diameter of about 70 microns. And the movement speeds that we were achieving are on the order of 10 to 20 microns per second, to give you an idea of scale.
Okay. So for the Brownian force, Brownian motion usually will happen below the 1 micron, right?
Right.
So
So there's
yeah.
Brownian motion is not a factor really at all here at this scale.
Yeah. So what's the driving force for the, for these droplets moving around, chasing around? What's the driving force behind that?
Right. So you have one of the droplets is dissolving and releasing a gradient of oil into the surroundings, and that gradient affects the interfacial tension on these droplets. So they move due to the Marangoni force that these droplets experience due to gradients and interfacial tension across them that are induced by the chemical gradients being patterned by these droplets.
Okay, so one is releasing the oil, another one is absorbing the oil. So that has to happen in a very fast and quick way, right?
So it's actually pretty gradual. So the solubilization rates of the bromooctane oil, which is the one that's being preferentially solubilized, is about 0.3, 0.4 microns per minute. So a 70 micron droplet will last for over an hour. So
I see.
yeah. But you can control the solubilization rate by the concentration of the surfactant as well as the oil type. So shorter oils generally have faster kinetics, and different types of surfactants have different kinetics associated with them as well.
Thank you. One more question. You mentioned that when the molecular of the oil droplets, I mean, the oil you use is very small, they actually soluble in water. How do you think like that? No matter how small they are, they should be insoluble in the water.
Well, so for that, those were pretty small oils. They were only a few carbons in length. So for iodopropane, it actually is somewhat water soluble, and because I mentioned the solubilization is relatively slow, the water dissolution pathway ends up being at least comparable in the order of magnitude to the micellar pathway. Yeah, there's the rates.
So I have a difficulty to understand this because, for example, acetone is immiscible with water. You think acetone is miscible with water?
Are they not miscible? I believe they're miscible, but that's not the oil. So you can't do this with sort of any combinations of oils. They have to be. You have to choose oils that are immiscible with your continuous phase to create an emulsion in the first place.
Okay, we can talk offline, but I have a very difficult to understand these points. I think that all the oil, no matter how small they are immiscible with water, no matter, unless you have a surfactant to help them to mix it together.
Yeah, I would think of it as a degree of how hydrophobic the molecule is, so and also its size. So as you get smaller and sort of less hydrophobic, you start to have chemical properties that have a finite and increasing water solubility.
Thank you very much.
There's a question in the chat box. Caleb.
Autochemotaxis. So are you referring to like the self-propulsion of these droplets? I think. So yes, to answer your question, yes, you can get these bromooctane droplets to become self-propelled, especially so the way you would do that is increasing the surfactant concentration. So if you increase it above sort of critical concentration, about 1 weight percent, these droplets would no longer chase the EFB prey. Instead, they would just sort of become self-propelled and would only be responding to their own gradient. So we're deliberately choosing experimental conditions below the sort of self-propulsion concentration in order to see this effect. Though you will notice in the video, like the smaller droplets tended to be self-propelled at lower surfactant concentrations. So you can see sort of that auto chemotactic movement happening for some of the lone droplets in this situation.
Caleb, I have one question.
Yeah.
Apparently these particles are very sensitive to, or the way they are propelled or interact, it has a lot to do with interface, right? Interfacial energy, surface tension. What happens if you put all these droplets on another interface, or liquid air interface, or liquid liquid interface? What do you think will happen?
So I've tried putting these at a water air interface. I think it might be possible, and like maybe the same sort of principle would apply if you could set up sort of a net exchange of chemicals somehow. That would create some sort of non-reciprocal dynamic, possibly. These particular oils would not do that, so you'd have to play around with your chemistry. I will say that there is a Nature paper by Prakash, I think 2015, where they showed that you could have like non-reciprocal interactions that are vapor mediated between sessile droplets of water and glycerol of different concentrations, and that would be a way to create a similar non-reciprocal dynamic.
Interesting. Any other questions? There's one more question from the chat box.
May I ask a question?
Sure, and then I'll take the chat.
Okay. Thank you for the nice talk. So for the mechanism of the propel of single droplet, it is, if I understand well, if my understanding is right, it's because of the gradient spontaneous break of the symmetry of the meso gradient for the single droplet. So when the two type of droplets has interact with each other and combined to one droplet, if I can say, so the mechanism is still the broken of the symmetry of the meso. So for this new type of droplets, for example, the speed compared with they are combined before. So how was the speed change? I mean, the combination of these two particles is facility or favor the motion or disfavor the motion of the particle.
So are you asking how does the speed of the chasing pair compare to the speed of like self-propulsion for like these droplets in the,
yes,
on the screen? Yeah, so they are, it depends, I guess. You can make a self-propelled droplet faster than a chasing pair if you just add more surfactant. So if you increase the surfactant concentration, you can get speeds of on the order of 1 droplet length per second, I would say, up to that. And as I mentioned, these droplets are like 70 microns, you're getting up to 20, 25 microns per second. So 1 droplet length every few seconds. However, I will a few things to point out. So one is that to get those faster speeds by increasing the surfactant concentration, your sort of efficiency is going down, right? Because you're releasing more chemical, you're spending more chemical energy to move. And there is a part of our paper where we do try and calculate the efficiency for some of these interactions between different oil pairs that you can take a look at. And the Janus droplets that I showed at the very end, we're getting really fast speeds from those that are like multiple droplet lengths per second. So orders of magnitude faster than the chasing pairs. So take a look at that if you're interested.
So thanks. But my what I want to ask is, like, for example, you have I don't know, you have isolated particle at first, the same particle A and the same particle B. What I want to know is after the combination of the two particles, like they have oil exchange between them, and they will, I know, they will have sort internal flow between these two droplets.
Mm-hmm.
I mean, like these, for example, like this internal frame, because of the combination and the oil exchange of these two particles, does it affect the swimming? Yeah, the mechanism of the swimming.
So maybe what I should have said is that individually, if you mix just one of these two droplet types under the same conditions, neither of them are active. So there's no self-propulsion happening for either droplet. It's only when you put both types of droplets together do you see any active behavior under these particular conditions. Yeah, so, but as I mentioned, that the bromo-octane droplets can become self-propelled if you increase the surfactant concentration further.
Yes, so in this case, the miscible concentration is not enough for the single isolated particle to self-propel, but when they combine, when the two particles combine to one, they start to move. So the move mechanism, is it the same as the one before?
Yes, it's still the sort of same, it's the same Marangoni mechanism, but the symmetry, the asymmetry introduced by having the two droplets helps it lowers the propulsion threshold is a way to think of it. Yeah.
Okay, that's what I'm asking. Thanks.
Great.
Thank you.
Caleb, can you very quickly comment on the two questions from Stephen and Ethan?
Yeah. So why is it? So it's to Stephen's question. So the way I would think of that is that you have the bromo-octane droplet, which is releasing a repulsive signal, and that repulsive signal is is modified by the fact that you have this droplet behaving as a sink. So the oil concentration between the droplets is lower than it is behind the predator, and so that's why the predator is attracted to the prey, and the prey is repelled by the predator because it's responding to the repulsive oil signal that it's releasing.
Remember, it's the micelle concentration that's different from the back and the front, right?
Oil-filled micelles, I guess, they are the surfactant and the oils are complexed in the aqueous phase, so you can sort of think of them as together. So the oil-filled micelle concentration is lower on this side than this side because the prey is uptaking some of the oil, and so that's what causes you to get this attraction of the predator.
And Ife was asking about the size.
Yes. So the size, it does matter. As I mentioned, the smaller droplets have a greater tendency to become self-propelled, and a chasing pair of smaller droplets moves somewhat slower. So yeah, as the predator droplets shrink in size relative to the prey, you will have a decrease in the speed of the pair.
All right, we're running short of time. I think that was a very good discussion and a wonderful presentation from Caleb. Thank you very much. And before everybody goes, I want to remind you that this talk has been recorded and we will upload them on YouTube for everyone who's outside of China. And for those inside China, we'll upload it to Bilibili. And so you can watch it later. All right, thank you so much. And thank you again, Caleb. And I'll see you again next week.
Awesome.
Thank you.
All right, thanks.
Thank
Bye-bye.
you for your presentation. Have a good
Thanks,
one.
everyone.